All right. We're going to move on to chapter three now. So chapter three is a really long chapter. So I'm just going to cover like the relevant parts and skip like the intro to cells, because that's more anatomy. I'm sure you guys went over that anatomy. If you guys haven't and need help, like there's something you don't understand, just ask me, and I can zoom with you for like 5 minutes and explain it all. Okay. All right. So now, uh, oops, slideshow. Okay, so the genome and the proteome. Okay, so I'm sure you guys heard these terms before, especially the genome, but I want to make sure you guys understand what these words actually mean. Okay, so your genome is actually all the genes in a particular individual. Okay, um, so it's like all your chromosomes put together, basically. Okay, uh, researchers believe humans have about 25,000 genes. The number is actually slightly less than that, about 23 is the last count. Okay. Not all genes code for proteins. Okay. Um, the one that code for protein is about 21,000. Uh, so some of the transcribers do different types of RNA and they do different RNA things, which we'll get into. Okay. The proteome is all the proteins that are produced by the genome. Okay. There are more than 100,000 proteins produced by the human body. Um, the cool thing is when we figured this shit out, we initially thought there would be 100,000 genes because there was 100,000 proteins. And it turned out we were dead wrong. Okay. And in fact, one of the things we're most dead wrong about is like we have the exact same number of genes as mice, rats, and other mammals. Okay. Even though, we would like to think of ourselves as somehow better or more special than they are, we have the same number of genes. What makes this interesting is genes actually can code for more than one protein. Okay. Okay. So how can a gene code for more than one protein? The mRNA is altered after transcription by splicing in different ways, and we'll get into that in a little bit. Okay. Proteins are made of many different polypeptide chains, and so you can have different combinations of them. Okay. Like A, B, B, like those different combinations all work. Okay. And honestly, some proteins have like 10 or 20 subunits and they can mix and match. Okay. Proteins can be modified by adding a lipid or a carbohydrate. That's less important than the last one, actually. Post-translational modification by methylation, you turn things on and off. Okay, phosphorylation, again, turn things on and off. Cutting into smaller units. The cutting into smaller units actually tend to activate the genes, okay, or sorry, activate the proteins. Okay. Okay. First thing, okay, I'm sure you guys can picture DNA being this ladder, that's a double helix and stuff like that. Okay. DNA in our cells is very, very, very rarely like that. Okay. Most of our DNA is protected by being packaged. Okay. The things that are packaged with are called histones. And so histones combined with the DNA form chromatin. Okay, histones are normally negatively charged and interact with the negatively charged DNA to cause spooling. Okay, they create particles called nucleosomes. So let's see what they look like. Okay, so these are nucleosomes. Each one of these four is a histone. Okay, and when DNA wraps around, it's called a nucleosome. Okay, this the non-scientific term for nucleosomes are beads on a string. You can literally see how they look like beads on a string. Okay, there are two types of chromosomes. Chromatin, sorry, euchromatin, which is active in transcription, is looser. okay? And then heterochromatin, which is the inactive region, that's where the DNA is highly inactive and just kind of locked up, okay? So there are chemical changes in the histones, such as acetylation, adding acetyl groups to the histones, which allow them to loosen up the chain, and it allows molecules to access DNA. The only time you can actually make DNA do something is you have access to the DNA. Okay, you can't make a protein if you don't have access to the DNA. So the heterochromatin, the DNA is going to be very inactive, and you're not going to get a lot of protein from. The euchromatin, that you are going to be able to transcribe genes from. Okay, one of the things we studied, or one of the things I studied, was different genes that modified heterochromatin and euchromatin, so you can get more like different proteins expressed. Okay. By the way, the gene was called SIR2, okay, silent information regulator 2. Okay. Okay. So here, okay, most of the DNA is actually in this format inside the cell. In mitosis, this is what happens. Okay, and we'll go over mitosis later. We form the actual chromosome later. Okay. 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 So here's the condensed chromosome. Okay, where you can't have any access. The acetylation causes this to form a different shape. Okay, which allows more access to the naked DNA. Okay, and then you have transcription factors. Okay, that can bind. Now, the regions are exposed, and they can bind transcription factors. And then the transcription factor, once it binds, it can unravel the rest of the histones off of it, and the polymerase can actually work. Okay. Okay. Anything you can activate via acetylation, you can deactivate via deaceleration. Okay. All right. DNA-directed RNA synthesis. Okay, also called transcription. Okay. So the way I think of this is, look, DNA and RNA are both nucleotides. So the language is nucleotides. Okay. So transcribing something literally means like copying. Okay, you're not changing languages. Translation is changing languages. We're going from DNA to RNA. Okay, so the way I explain this usually is DNA is a library. Okay, the library has to get the books inside the library are the genes. Okay, the books have to stay inside the library, but the only way you get information out from the library is to take it in, copy by hand whatever you want, or photocopy whatever you want, and then take that photocopy out. Okay, that's the RNA. Okay, okay. The start and stop regions at the beginning and end of the gene. Okay. Right ahead of the start, okay, is usually things called promoters. Okay, promoters are areas of DNA that are not part of the gene but tell the enzymes involved where to begin. Okay, so if I took a gene and I chopped off the promoter, no gene transcription. If I took a gene and put on a really active promoter, a lot of gene transcription. Okay. And then the transcription factors bind to the promoter, okay, and begin transcription. Okay. So transcription factors are little proteins inside the cell that tell the cell which particular genes I want to transcribe. Okay, so what makes a muscle cell different than a brain cell is that the muscle cell has has transcription factors. Okay, for muscles, the brain cell has transcription factors for brain. Okay, the DNA is actually the same. Okay. RNA polymerase. Okay. Is the enzyme involved. Okay. The RNA polymerase is exactly what it sounds like. It makes RNA and it's a polymerase. So it makes a RNA polymer. Okay. So breaks the hydrogen bond between the pairs of DNA and assembles the appropriate RNA nucleotide. So there's a strand of the DNA is in two strands. One strand is called the template strand. Okay, the other plate is called the non-template strand. Okay, the RNA nucleotides pair up to the DNA template. Okay, so if the DNA template strand reads GCTA, then the RNA will read CUA or CGAU. Can we read now? All right, so RNA has uracil instead of thymine. Okay, so there's no T's in RNA. Okay, it forms precursor messenger RNA that detaches from the RNA or DNA template. Okay, so only one free strand of DNA is transcribed. Okay, okay, okay. There are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. The precursor messenger RNA is the one that's going to be initially made, but it's going to be cut into mRNA right away. Okay, and I'll explain what that is. Okay, so the sequence you originally come up with, the pre-mRNA, is not actually the sequence that leaves the nucleus. Okay, there are portions of the gene that do not code for proteins, and they're called introns, and they get cut out or spliced out. Okay, so the portions that are kept are called exons because they're expressed. Okay, so the way I remember which one's which, introns stay in, exons go out. Okay, exons exit the nucleus. Okay. Alternative splicing allows one precursor mRNA to make multiple proteins. Okay, so the exons are joined together by spliceosomes and SNRPs. Okay, which are small nuclear riboproteins. Okay, so here you can see the DNA here. This is the RNA. Okay, once you make it, this is the pre-RNA, and then you cut out all the introns, okay? And you can splice them together, okay? The way splicing works, the way you can make more than one protein, okay? One, two, okay. Just the boxes are exons, the lines are introns. Okay, so this is one, this is two, this is three, and this is four. Okay, so I can go, so write this by hand. I can go 1, 2, 3, 4, or 1, 2, 3, or 1, 3, 4, or 2, 3, 4. as long as the numbers are in order. I can't go like 2, 1, 3. Okay, but anything that's in order, 1, 3, 4. Okay, that's fine. Okay, so I can, from just those 4, I can make, well, you can already see I can make a lot of combinations. Okay, and some of the bigger genes have something like hundreds and thousands of introns. Okay, so you can slice them. I think there's one gene in the fly which had like something like 100,000 different combinations of splicing. Okay. All right. Clear. No. Okay. Okay. RNAi or RNA interference. It's so cool to be teaching you this stuff because when I was in grad school, this shit was like brand freaking new. Like we couldn't believe that this actually occurred. Um, and the story behind it is actually really interesting, but I don't have time to tell it. Okay, so RNA molecules that don't code for proteins may prevent some mRNA molecules from being translated. Okay, um, there are 3 types. Okay, short interfering RNA, siRNA, miRNA, and circRNA or circular RNA. Okay, so the expression of at least 30% of genes is regulated this way. Okay, so small interfering RNA is formed from a larger double-stranded RNA molecule that leaves the nucleus. Okay, it attaches to a enzyme called dicer. Dicer does what you think a dicer does. It chops things into pieces. Okay, the 21 to 25 nucleotide long fragment then can attach to pieces of messenger RNA, and then that gets chopped into bits too. So if the messenger RNA is chopped into bits, then the signal, there's no protein made. Okay, so small interfering RNAs ultimately prevent genes from being expressed. Okay, so you can actually turn off genes by expressing the siRNA. Okay. Okay. mRNA forms a single-stranded single-strand RNA that forms a hairpin. Okay, a hairpin. I'm assuming they show you guys a picture of a hairpin. They'll show you a hairpin. Okay. Let me show you guys a hairpin. So here's the RNA strand. Okay, the RNA strand will go into a loop and hairpin bind with itself. So there's an A there that will bind to a U there, a C there will bind to a G there, and C, G, etc., all the way through, and that will form the hairpin loop. Okay. Okay. The hairpin will then look like a double-stranded RNA, just like the siRNA. That will be again cut by dicer into these double-stranded segments, and then it will do the same thing. Any mRNA that matches those segments will be prevented from gene expression by cut dicer, because dicer will cut that in part too. Okay. 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 So circular RNA. This is so mRNA prevents the transcription of the original mRNA. circRNA prevents the prevention of that. Okay, which is one of the weirdest things ever. Okay. So within a risk particle, hold on 1 second. Sorry about that. Let me go back to risk. Okay. So within a risk particle, okay. So this concept of small siRNAs and mRNAs silencing genes is called RNA-induced silencing. Okay. Okay. So it's called a risk complex. This RNA-induced silencing complex. Okay. This certain RNA interferes with the complex, and therefore it prevents silencing. So off. You turn off the off switch. You turn the cell machinery back on. Okay. Okay. So there's the risk complex. Okay. Okay. And the technique is called RNAi or RNA interference. Okay. RNA interference may potentially help in cancer treatments as well as other genetic conditions. If you have a genetic condition where you're expressing a gene that's bad for you, if I can use RNAi to turn it off, you're back to normal. Okay. So cancer genes, cancer cells express a lot of genes that promote cell growth. If I can use RNAi to turn off those genes, you slow down cancer growth. Okay. All right. Even cooler is the CRISPR-Cas9 method. Okay. This can lead to direct gene editing, which is actually really cool. Okay. So this is new as of like 10 years ago. So even after I finished grad school. Okay. The CRISPR-Cas9 is a unique sequence of bacterial DNA on either side of some target of gene of interest to scientists. Okay, so whatever I put in the middle of this sequence, I can target. Cool. So now I can specifically target whatever gene I want. I just put that gene I want in between these palindromes, and there we go. Okay, the target enlists the function of a gene called Cas9. Okay, Cas9. Cas9 is a gene that normally destroys DNA. It's usually turned on when the cell goes into apoptosis. Okay, when the cell commits suicide. Okay. So now I directly get Cas9 to cut exactly where I want to cut. Okay, so I can remove a specific gene. Okay. 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 So scientists can use CRISPR-Cas9 system modified in a different way to activate instead of repressive genes. If I deactivate the deactivator, then I activate the gene. Okay, so all genes have activators and things called repressors, right? So if I deactivate the repressor, I activate the gene more. Okay. All right. I'm gonna stop there. I'm gonna pick this up in part 2. Okay.